This is going to be an experimental analysis of our project, vitamin C concentration and the effects of pasteurization. As an introduction, vitamins are organic compounds essential for cell growth, function, and development by facilitating chemical reactions in the cells in a catalytic nature. Though necessary to the living organism, vitamins are not synthesized in the body and therefore must be obtained from the diet in small quantities. Vitamins are either water-soluble, which are used up immediately by the organism and the excess is flushed out, or fat-soluble, which are stored in the fatty tissues of the body. Vitamin C is water-soluble, and when digested, enzymes in the body facilitate in the breaking down of vitamin C by lowering the activation energy required for chemical reactions to take place. In humans, vitamin C is an antioxidant, an essential vitamin for skin, bone, and connective tissue, as well as a key component in the absorbance of iron into the body. A deficiency in vitamin C causes a disease known as scurvy. Symptoms of scurvy can include bruising more readily, bleeding of the gums, and anemia. Vitamin C is best obtained in its raw form, as in its cooked form, the heat can denature the ascorbic acid. Because heat can denature vitamin C molecules, it is important to understand the effects that pasteurization can have on the vitamin C concentration of orange juice. So we hypothesize that if orange juice is pasteurized, the vitamin C concentration will decrease because the increase in temperature will cause the covalent bonds within the molecule to break. So for our experiment, a single orange was cut in half and both halves were juiced on a plastic juicer. The juice from each half was combined and strained through 4 sheets of cheesecloth into a 250 milliliter Pyrex beaker. The pulp caught in the cheesecloth was then discarded. This was necessary because the presence of its sediment can interfere with the readings of the spectrophotometer. 3 replicates were performed for each of the 2 sample sets, the pasteurized and the unpasteurized. 3 replicates were performed because that is the minimum number required to identify any potential outliers. Each replicate was taken from the same sample of orange juice extracted in the previous step. 1.5 milliliters of the strained juice was then pipetted into appropriately labeled microcentrifuge tubes for centrifugation. Prior to centrifugation, the samples were placed in an ice bath for 2 minutes. After the 2 minutes in the ice bath, the microcentrifuge tubes were placed in the centrifuge and centrifuged at 12,000 RPM for 2 minutes. The centrifugation is necessary as it filters out any remaining sediment that was not successfully filtered out through the cheesecloth. While in the centrifuge, additional microcentrifuge tubes were labeled identically to the first group to transfer the supernatant into. Samples were taken out of the centrifuge and, careful not to disturb the pellet, the supernatant was transferred into the newly labeled microcentrifuge tubes using a pipette. Samples D, E, and F were placed back into the ice bath during the pasteurization of samples A, B, and C. The microcentrifuge tubes labeled A, B, and C were placed in a floating rack and left in a water bath at 93 degrees Celsius for 2 minutes. After 2 minutes, the floating rack was removed from the 93 degrees Celsius water bath and placed in another water bath at 80 degrees Celsius for 20 minutes. The floating rack was removed from the water and the microcentrifuge tubes were placed in an ice bath for 5 minutes. So for our experiment, we used an organic dye known as DCIP that when in a basic solution is blue and when reacting in an acidic solution is red or pink, followed by fully clear. Vitamin C affects the color and in turn the amount of light able to pass through the cuvette. Vitamin C loses 2 hydrogen atoms in a process called oxidation, and DCIP gains those 2 hydrogen atoms in a process known as reduction and turns to DCIPH2. DCIHP2 is clear and will register as a 0 absorbance at 520 nanometers, as compared to a registerable reading with DCIP. A standard curve can be created as differing levels of vitamin C will react to the DCIP, creating a range of colors from red, pink to clear. Once the curve is created, the vitamin C concentration can be estimated, finding the absorbance of a specific sample. Prior to testing, the dilution ratio was calculated to be 1 to 5. The dilution is necessary because too much vitamin C will force a reaction with all the DCIP molecules, turning them to DCIPH2, producing a clear solution. The dilution will make it so all the samples have readable results. Adjustments can later be made to compensate for this dilution. The following measurements were used: 500 microliters of DCIP solution, 980 microliters of distilled water, and 20 microliters of juice. Samples A through F were diluted using distilled water and DCIP solution in a cuvette using micropipette for measuring. A piece of paraffin wax was pinched over the top of each of the cuvettes, and the cuvettes were inverted several times to thoroughly mix each solution. The spectrophotometer was then adjusted to 520 nanometers, and each of the samples was placed into the spectrometer, and the absorbance of each sample was recorded. The absorbance was then multiplied by 5 to adjust for the dilution. So as you can see in figure 1, we have a picture of the standard curve that we used to estimate the vitamin C concentration. This shows the usable values. Several values had either too little vitamin C, which would not react enough with the DCIP to change the color, or too much, which would react to produce a clear solution, and the difference would be unmeasurable. As outlined before, this was calculated prior to class and shows the line of estimate for the relationship between vitamin C concentration and absorbance. Using the absorbance levels we found, we can use this graph to find the vitamin C concentration. Shown in figure 2, we have the vitamin C concentrations of both the pasteurized and the unpasteurized samples. So the average vitamin C concentration of the unpasteurized orange juice was found to be 1,911 micrograms per milliliter, while the average of vitamin C concentration of the pasteurized orange juice was found to be 1,684 micrograms per milliliter. The difference between the average vitamin C concentration of the pasteurized and unpasteurized samples is 11.8%. Important to note, within each sample there is also some variation. The pasteurized samples show a 4.9% variation, while the unpasteurized samples show a 4.7% variation. Because the percent variation within each sample set was so much less than the percent difference between the fresh and unpasteurized juice vitamin concentration, it is unlikely that the difference was due to chance. It was hypothesized that the pasteurization would lower the vitamin C concentration of orange juice, and our data supports this hypothesis. Also important, the design of the experiment and execute it proved adequate to test our hypothesis. When examining the results of other groups, we see a similar trend, though the Love group had data most similar to our own. The Love group found an average vitamin C concentration of 1,879 micrograms per milliliter for the fresh juice and 1,668 micrograms per milliliter for the pasteurized juice, an 11.2% difference, but showed 13.5% variability amongst the fresh samples and 10.1% variability amongst the pasteurized samples. The Bowling group found an average vitamin C concentration of 1,349 micrograms per milliliter for fresh juice and 1,434 micrograms per milliliter for pasteurized juice, a 5.9% difference. But they showed a 48.6% variability amongst the fresh samples and a 14.6% variability amongst the pasteurized samples. Finally, the Padilla group found an average vitamin C concentration of 2,021 micrograms per milliliter for fresh juice and 1,999 micrograms per milliliter for pasteurized juice, a 1% difference. But within each sample showed a 13.6% variability amongst the fresh samples and a 10.5% variability amongst the pasteurized samples. Important to note, each of the other lab groups had one case where the percentage variability amongst each sample set was higher than the percent difference between the sample set. In each case, this was due to a single outlier that skewed the results in one direction or another. These outliers, as well as the variability amongst individual groups, could be because of a variety of reasons. Different group members were all having to pipette, and the different techniques employed by them could be a source of variation. Possible leftover in the sediment, possible sediment leftover within the sample, faulty measurements, or the instruments themselves could also be sources of some variability. To improve this experiment in the future, additional replicates could be helpful as they could produce a more accurate average and weed out other outliers. And lastly, using a centrifuge with the capacity to spin faster would also be helpful, as it would ensure that no particles were left behind to disturb the spectrophotometer readings.